Good afternoon, everybody. It's a pleasure to see everybody out back at conferences again, and love to thank ARM for having us and allowing us to introduce you all to Forge Biologics. So a little description of the company. Forge is we look at ourselves as a hub and spoke model where we're looking to produce AAV as a CDMO, and we're doing this in an end-to-end fashion with both development services, analytical, and production services, which I'll get into a little bit about our scale as the majority of our business. But we also carry a therapeutic pipeline as well, where we're developing rare disease AAV therapies, and there's a lot of advantages for that as well. Not only are we able to produce these rare disease pipelines for patients, it also gives us an opportunity to develop a lot of our CDMO tools and move them in front of regulators and really get feedback before we start working with our clients. So we developed this program and this company, and in the last 18 months have really made a lot of advance. And I can speak for the team that it's been a really rewarding 18 months and what we've accomplished and really how we can move ourselves along. And we did that through a series of 3 financings that raised about $240 million in the U.S., successful completion of the build out of a GMP facility. And we all know the difficulties and the trials that run through that, but also moving through the qualification for AAV manufacturing, establishing our cell lines for our clients' use. And I'll talk a little bit about the tools that we offer exclusively at Forge for their use as well, which also includes the regulatory advantages, and integrating what we call our Blaze research production. This is an off-the-shelf, sort of think of it like a viral core way to come in and try out working with Forge and really look at some of your new constructs and say, really, what are some of the initial data and titers looking like off of our options here? And we can work with you in order to help you make the right selections for your downstream candidates. We've continued to advance our own clinical pipeline. We have one that's in the clinic now and one that's under development. And, excitingly, our team has grown quite large in just 18 months. We're already at over 190 employees. The majority of those are in our development, both in product development, process development, analytical, and also our GMP and quality areas. So really excited to see the team expand. And, I think it really comes down to the leadership team, which has about 165 years, a little over, of gene therapy experience. We were able to collect a great array of experts to come in and really help us develop this. And what you'll notice is the majority of folks were formerly developers of their own. So worked with CDMOs, really know the development process, and really try to integrate that field back into how we work with our clients and the way we can advise them through the CMC process. So we're led by Dr. Timothy Miller. He's our CEO. He was formerly with Abeona Therapeutics and has a long history in the cell and gene therapy space. Just some folks to point out: Jason Eichholz, formerly with the Nationwide Children's, ran the core there. Randy De Silva in the area with Maria Escolar as our CMO. I'll talk a little about our clinical pipeline. David Dismuke, who is our CTO and really driving a lot of our innovative tools and platforms and really advising our clients through their development process, and an array of others in business and also finance and also our clinical space. So really excited to collect this team together, and really think really drives a lot of the confidence from clients. So talking a little bit about, why we're focusing on AAV, and we thought about, there's obviously you can work broadly and work in other and other viral vector types, but the AAV space we saw not only with the expertise on the team, but also the growth in the space over the years that we projected. We performed this independent third-party analysis of what does the sort of field look like going out in 2026, and I think as a lot of us probably have realized, the growth in the clinical side, first of all, is quite broad. As you can see here in this analysis, the move into not only expanding phase 1 and phase 2 trials, but I think the interesting part will become the phase 3 trials, which, as we all know, eventually we hope to see become more commercial solutions for patients. That demand from a bioreactor volume standpoint really increases sharply. And, the solution to that we were thinking about is really focusing on AAV, but also can we build one of the largest AAV facilities in the world? And that's our goal, is, we really are trying to look at what sort of demand can we meet. And if you think about larger disease states like DMD and hemophilia, the yield requirements and the title requirements for dosing are quite high, and you're talking about patient populations that are growing, and this is before even starting to talk about non-rare disease, which obviously is the ultimate goal for gene therapy. So where we are today to now is an assessment, again, by the same independent study we've done is, what Forge believes it can take as far as the overall capacity, world capacity of AAV manufacturing. And I'll share some of our footprint shortly, but we believe with our plan, we can be looking at taking nearly, up to 22% or more of the future expansion of the actual per volume liter bioreactor space in the world. So obviously that fits our goal of being the largest and the sort of the leader in that area. That also comes with the demand, and I think the demand does meet that need. So we really, goal was to partner those together tightly. So what are we offering? So everything from end to end is really our goal, and we talk about from initiation to injection, we talk about gene in a dream, really. The goal was to create a platform process that you can plug in your GOI and then really develop that straight through and have a really robust system that's across multiple serotypes, or it can accept novel capsids and really accelerate our clients' pathway to development. And obviously from a growth and financial standpoint for small companies, especially, that sometimes is of concern. So we address process and purification as part of our packaging for what we offer, including fully disposable single-use systems, optimizing for purity and potency, and really establishing your process both upstream and downstream. Our manufacturing process and GMP plant is operational now and expanding. I'll show you shortly. We're actually moving from 50 liter to 5,000 liter capacity for GMP. A lot of that is driven by the demand of driving down costs commercially as well. Obviously larger batching and so is really ideal for customers. Our facility is 200,000 square feet. It's located in Columbus, Ohio, in the United States. Pretty ideally located for distribution as well, especially with its proximity to airports in the area around the US, but also in Europe. And, really full supply chain, warehousing, cold chain, freezer farm, all done in-house, including full analytical capabilities. And then, of course, our full R&D. And I talked a little bit about our off-the-shelf offerings, but we can perform 1 to 50 liter research and grade tox production runs. This can be for early stage preclinical, for your CMC analysis, and obviously up to your GLP studies. And then we have multiple tools to help you along the way with this and in-house development. Our analytics, our goal is to bring everything in-house. We have most of that done so far. Sterility, mycoplasma, things like adventitious agents would probably be things we'll continue to outsource, due to controls. But everything else down to RCAV, AUC, that's something that all will be on-site at Forge for testing needs. So talk a little bit about some of our specific offerings that we give. These are Forge technologies that were developed in-house that we think have become actually quite popular offerings with our clients because it gives them some advantages not only from a purity and a safety standpoint, but also from a regulatory standpoint, which I think has become very key in the discussion around AAV about really, what are the right ways to sort of discuss the history of a lot of the tools that you use. And I mean that specifically for what's called our ignition cells. This is our suspension HEK293 mammalian cell line. It's been developed in-house at Forge. It's a clonal cell line that we offer to our clients. And the reason why we think it's important is it actually has a very well-defined history. And I think if you've worked with certain cell lines in the past and found you've had some black boxes around some historical portions of the cell line, it leads to questions from regulators. And our goal there was to create a cell line that goes back to the original license for the HEK cell lines and is in our hands as a 5-year renewable that we can sub-license to our clients. That gives you, unfeathered access to what the history of that line is. How do you actually refer that back, and through a DMF that we're going to offer all of our clients. It's already submitted in the US, and we'll have something similar in Europe as well. So we think it's an important component of our offerings, animal component free, and obviously suspension and scalable as we're thinking about going beyond 500 liters to 1,000, 2,000, and 5,000 liters. We also offer the P-Ember, which is our helper plasmid. This has been developed compared to other commercially available helper plasmids to reduce the size by eliminating some sort of unnecessary target regions that are potentially immunogenic or fiber regions that are not required, and actually makes it a bit smaller as well, which is good from a cost and an efficiency standpoint. So this is exclusively offered at Forge from our clients as well, and is available for licensing. We do these royalty-free, so we don't look to hold you as a sort of a hostage and looking for royalties in the future. You work with us and perform a GMP run. These would then be licensed to you to either obviously stay with Forge, or if you're looking to do in-house manufacturing or move elsewhere, then that asset can be given to you after as a licensed final GMP run. Blaze production, again, I touched on it earlier, but this is our chromatography-based, off-the-shelf. Certain assays come with it, and this isn't sort of product development. This is process development. This is you being able to try out working with Forge, try out your constructs, collect some early data, and start making some real decisions about what you want to carry downstream. We think this saves a lot of time too for clients up front, because they get to do that selection rather through a lengthy development process. It causes them to eliminate certain promoter options, certain elements and constructs. It really narrows it down in a good way for them to move forward in an efficient manner. So here's a quick snapshot of the hearth. This is our facility in Columbus. It's a 200,000 square foot shell, and then we are building our capabilities under that shell. And what exists today is what you see more on the left side of the image. We have what we call our Stage 1 GMP Manufacturing Center. This is a Phase 1-2 ready manufacturing center. It's operational today. We are producing GMP materials in it as we speak. And it's designed and geared, we want to get it up and get people moving early so they can start delivering on their clinical trials. We also have active PD and AD space. This is through all of our development work that I mentioned, all of our analytical development as well, and then your tox material can also be produced in CNC space in this area, or obviously you have the GMP space for engineering and GMP future runs. What we're doing now is we're in the midst of constructing our Stage 2 portion of our facility. That is the part you see in the middle there, and actually includes the PD expansion area and then some warehousing, cold chain, and also other support areas that will support not only for this future expansion. This is an additional 16 rooms of capacity designed for EU standards, and also will contain 5,000 liter bioreactors. As I mentioned, we'll have 2 of them in the space, and then also automated filling and for fill finish. So this will be our commercial ready space as well. So as these clients are moving through Phase 1-2 and Phase 3, we can continue to carry them all the way through into commercial as well. So really excited about this, looking to have that in the second half of the year, complete in construction and through its EMPQ work, and already have clients signing up to join this space for the use going into 23. As you can see, there's still a lot of gray space for us to do future expansion, so we can continue to add suites, and we also have some flex space as well. So excited to keep with the pace of the industry on that front. There's a video on our website I'll point you to for forgebiologics.com. You can see some time lapse of the build. I think it's really cool stuff to see a lot of it going up, especially HVAC and then walls going up. We really enjoy sharing a lot of this information. So we have a lot of videos like this that are on our website to check out about our progress. And then working in Europe is a second half endeavor. We're starting now, and with the med meeting here and establishing our presence. We're going to have client development actually in the EU, likely working out of Spain or some other areas as well. And our Stage 2 facility, as I mentioned, EMA compliant from a background standpoint and full fill finish. Our cold chain development for shipping over here for Europe, and also offering all of our capabilities, for our European partners and our QP activities. So all this is ongoing. We're engaging with folks here in the EU and other regions around the world, and able to provide that and roll these plans out coming up in the second half of the year. So that's our CDMO business. In the last minute or so, I'm going to touch on our therapeutic pipeline. And I think it's important to touch on because this also not only is important from a patient standpoint, but also what drives a lot of these tools as well. So the tools that I mentioned have been in front of either FDA or EMA and had feedback. So that, what we like about these programs also is that it gives us the opportunity to review those. So we have 2 pipeline items, including infantile Krabbe disease, which is currently in phase 1/2. And then we have a second rare pediatric asset we have not announced yet, but looking to continue to build this pipeline through our business development activities. So the Krabbe program is led by Dr. Maria Escolar, who is our CMO, also Dr. Juan Ruiz, who is our head of clinical affairs, are running these programs. And basically Krabbe, just to give you a quick overview, it's a leukodystrophy. It's a rare neurodegenerative degenerative disorder. Impacts children from birth into about the age of 2 when, unfortunately, premature death is common. It's a terrible disease. It's a monogenic disorder from the GAL-C enzyme and leads to demyelination and neuron death. So obviously it's a huge issue impacting both the CNS and the peripheral nervous system as well, and obviously a rare disorder. Our approach is an AAV-RH10, where we're actually, the uniqueness of this approach is that we're combining this with hematopoietic transplant and doing a dual treatment in order to help these kids. And as I mentioned, currently in phase 1/2. Last slide, just a little bit of the preclinical data. In the Twitcher mouse model, we've seen, obviously an effect from either transplant or AAV, but the combined effect showing the best outcome for not only survivability, but also weight and some other sort of neurocognitive activities as well. So we're really excited about these data. We also have some dog model data that was really positive. So in the clinic now and looking forward to presenting some data in progress in the near future. So great combination for the company and really looking forward to giving you some more information on our progress. So thanks very much. I appreciate your time.